All right, welcome back, everyone. So for our next talk, I'm pleased to introduce Joe Makowiecki, who's the Director of Business Development Enterprise Solutions at Cytiva. He has over 25 years in the bioprocess industry, and Joe's going to tell us about the challenges and solutions to the rapid deployment of mRNA manufacturing. So over to you, Joe. Thank you.
Great, thanks a lot. Okay, thank you, everyone. So again, my name's Joe Makowiecki, and I'm the Director of BD for Cytiva's Enterprise Solutions. So I'm responsible for our end-to-end biomanufacturing platforms and modular facility solutions. So I'm really happy to be here today talking about rapid deployment of mRNA capacity and the importance of flexibility and modularity and end-to-end solutions. I'm also quite happy to be here and presenting along with our newest Danaher colleagues from Precision Nanosystems as well as Vanrex. So I'll start off with a little bit of an introduction on mRNA, talk about some of the manufacturing considerations, and then go into solutions and do a wrap up with a bit of future outlook and summary. So let's take a look at the current state of mRNA manufacturing. So vaccine development, as we have known it, takes several decades from the identification of a pathogen being linked to a disease to a licensed vaccine, with traditional vaccines often stretching out past a decade from development to market. And obviously with the urgency of COVID-19, we found many new ways to accelerate, and Pfizer and BioNTech, as well as Moderna, shattered that conventional development to timeline market. So what was different this time? There were many collaborations formed between academia, research companies, and established manufacturers. There was significant funding put into the evaluation of new vaccine technologies such as mRNA, as well as viral vectors. And these new vaccine technologies really enable that speed to market needed for pandemic response. So the regulatory agencies, they didn't have any less rigor, but they created a fast track for the COVID vaccines, enabling acceleration in the review and approval processes. There was also significant investments that were made on increasing manufacturing capacity in parallel to product development and clinical trials. The pandemic, however, did also put a spotlight on some of the weaknesses in our society, particularly around logistical challenges and the dependency on outsourced manufacturing rather than self-sufficiency. So this led to many countries reevaluating their in-country, for-country vaccine manufacturing strategies. And however, even when you have vaccine manufacturing capabilities, you still may face some challenges. So as all of us know, the mRNA technology is quite new to many. There aren't always solutions available at scale that are specifically designed for mRNA. So you may have to use technologies that are developed from other molecules or developed for other molecules and adapt them to your mRNA manufacturing. You may even have to rely upon homegrown solutions. Many manufacturers don't have the capabilities such as the infrastructure, the equipment, the knowledge, and the experience to even accommodate mRNA manufacturing. And for those that have capabilities, many times the workflow, it's scattered and distributed across countries, maybe even continents. The number of suppliers of critical raw materials in this space is limited. The materials needed for mRNA manufacturing may not be of sufficient quality, or the supply may not be able to keep up with the demand. So these logistical and supply chain challenges can become bottlenecks in the mRNA manufacturing space. So what manufacturing scale would you need to produce a vaccine for the entire world? What's a reasonable manufacturing scale? How many manufacturing lines would be needed? So this is a table. It's a high-level example showing the yearly output of a manufacturing line at different in vitro transcriptions scales. So this is assuming an average about 5 grams per liter production and about 55 manufacturing batches per year. So this means a little bit more than one batch per per week. We also have in this table different process yields. So now let's look at the 50-liter IVT scale, which seems to be the focus of several of the mRNA manufacturers today. So for the two currently approved mRNA vaccines, you would expect an output of anywhere between 55 to 275 million doses a year, obviously depending on the yield. So with the expected output of a 50-liter IVT scale and assuming either a 30 or 100 microgram dose, you would need anywhere between 30 and 145 manufacturing lines to produce the 8 billion doses in 1 year. That said, if we look at self-amplifying mRNA vaccine, where the expected dose is anywhere from 300 to 1,000-fold lower compared to the current mRNA vaccines, you would achieve the 8 billion doses from just one manufacturing line. So if we are to vaccinate the world, in a very short time, mRNA is a promising platform. So mRNA doesn't have to be large scale. However, for the current pandemic and for the potential future vaccines, it would be desirable for many to have a manufacturing platform that is flexible, that is fast, and localized. So now let's look at some of the points to consider on mRNA manufacturing. So some of the considerations in mRNA manufacturing are related to the molecule itself, the size. mRNA is a large molecule, typically 10-fold larger than the protein of the same molecular weight. It's not just a long string, it's loosely folded into loops and has three-dimensional structure. And the structure will impact safety as well as efficacy. So the 3D structure of an mRNA molecule, it will determine its function and will impact translation. And double-stranded RNA can trigger an immune response. So therefore, you want to reduce the amount of double-stranded RNA in your product. And then around stability, we know that mRNA has limited stability. And the limited stability of the mRNA will highly impact manufacturing, possibly requiring reduced processing times, cold processing, and cold storage. And we know there are ways to protect and stabilize it, obviously through the poly A tail and the molecule capping, as well as the encapsulation into lipid nanoparticles will serve as a delivery vehicle, as well as it will improve molecule stability. So as we look at the mRNA manufacturing process, it really consists of three different segments. It's the plasmid manufacturing, the mRNA manufacturing, and the encapsulation. So the plasmid manufacturing process, it is a microbial fermentation process utilizing E. coli. So after fermentation, you have cell concentration, you have cell lysis, precipitation, flocculation steps. That's followed by a clarification step. And then the downstream process starts with tangential flow filtration, followed by several chromatography purification steps, and ends with a TFF step. As we look at mRNA manufacturing, this process is a cell-free enzymatic synthesis. So the plasmid DNA is linearized, and then it serves as a template for the mRNA synthesis. So enzymatic capping of the mRNA is performed to improve stability as well as promote translation. Following synthesis and capping steps, the downstream is a series of TFF as well as chromatography steps. As we look at the encapsulation process, it involves the encapsulation of the mRNA into a lipid nanoparticle. And this step utilizes high concentrations of ethanol as well as lipids. And then after encapsulation, dilution is required to stabilize the lipid nanoparticle. So following encapsulation and dilution, a TFF is typically performed. I have seen in some cases that customers are using chromatography in addition to TFF, but mainly it's a TFF and you're buffer exchanging the product to remove any remaining solvents and getting it ready for drug product filling. So let's look at some of the considerations and strategies for manufacturing. So from an mRNA IVT reaction, Consider the availability and the quality of the reagents as well as the template DNA. Consider the efficiency of the IVT and the capping strategy used. Consider the expected mRNA titer, because the titer, the capping strategy, and efficiency will determine how you design your downstream. And also consider avoid generating, the impurities such as the double-stranded mRNA, as that will allow for a more simplified downstream purification strategy. On the purification side, consider the removal of impurities generated. So this is the double-stranded mRNA. This is the apparently capped versions, the runoff transcripts that can be of different length compared to the product of interest, as well as the residual reagents, the nucleosides, the enzymes, and the template DNA. Keep in mind that many products currently on the market today are not specifically designed for mRNA cGMP manufacturing. And we talked about the challenges of mRNA itself because of its size and shape. And there may be traditional purification technologies that may not be effective or may not be efficient at this point in time. You should always think scalability. Some methods and solutions being used for removing, for example, double-stranded RNA utilize solvents. They utilize elevated temperatures, which may work well at the bench scale, but may be challenging as you start to scale that process up. And think about flexible manufacturing equipment. It is available and can be adapted to accommodate mRNA manufacturing. And also think about automation and process control strategies, across that can be adapted across the mRNA manufacturing ecosystem. And it's used for improving the process monitoring, understanding, as well as data collection, and will help to reduce batch variability. As we look at encapsulation, obviously this is a critical step. It can have a lot of variability. There is limited industry knowledge and experience with encapsulation, and there are limited available and compliant encapsulation technologies on the market today. The encapsulation utilizes solvents, so you need to ensure that your equipment as well as your facility are compatible and they're appropriately designed to handle solvents. And when you're selecting a supplier, you make sure you have a provider with experience, with expertise in this space, as well as a supplier that offers a broad level of supporting and enabling services, such as process development, scale-up, cGMP manufacturing, technical transfer, and training, because that will make your processing much easier. As we look at drug product filling, mRNA manufacturing has a significant output, so you can get millions of doses from each batch. Many manufacturers currently have to outsource filling as they lack the internal filling capabilities. Limited stability of the product can generate challenges for the filling of the product. And make sure to size the filling capacity by scaling up or scaling up. Scaling up may be favorable if you have only one large product, but if you have multiple products at multiple scales, a scale out strategy may accommodate a lot better. Flexible, standardized, single-use, enclosed, and rapidly deployable filling systems are available on the market today. These systems will easily enable in-house filling capabilities. So as discussed in the mRNA workflow, so there are several segments of the process that you can choose to either insource or outsource. This applies, for instance, to plasmid manufacturing. So the benefits of insourcing, obviously, there's self-sufficiency as well as control. You have control of the process, which includes access to and quality of the plasmid manufactured. You will have the plasmids when you need it and at the appropriate quantities. Also, you will have better control over the costs of manufacturing. So if you decide to insource your pDNA manufacturing, so what scale should you target? Since each pDNA will be read several times in the IVT process, and similar to mRNA manufacturing, you don't necessarily need large-scale plasmid manufacturing line to support the manufacturing of mRNA. So this table shows an example of the quantity of mRNA needed to support 100 million doses of the vaccine, as well as the quantity of plasmids needed to support mRNA manufacturing. And we've made some assumptions here, obviously. So the gene of interest is around 400 nucleotides. Each plasmid would give about 300 copies of mRNA. The plasmid manufacturing line will produce around 55 batches per year, and then the mRNA dose is around 50 micrograms. So it would require about 30 pDNA batches at a scale of 50 liters to support the generation of 100 million doses of mRNA. So later in this presentation, I'll show an example of what plasmid manufacturing solution could look like. So flexibility is key. It's always important to invest in flexibility, especially in an industry where we see quite a lot of product diversity. And many of these new product modalities are rapidly evolving as processes, as products to support them are also adapting and they're also improving. So a flexible platform will enable adoption to new products, new processes, as well as new scales. And modeling is key. It's important to model different manufacturing scenarios as that will guide your flexibility requirements. And by modeling different scenarios, you can ensure that the core equipment will meet your desired product, desired process, and scale requirements. So the greatest flexibility is typically achieved through the utilization of single-use equipment. So modeling will also help guide your scale strategy, whether you decide to scale up or whether you decide to scale out. It will also help to identify a lot of the bottlenecks that you may encounter along the way. So think flexibility and optimize manufacturing utilization for your investment. So let's take a look at some of the manufacturing batch scheduling scenarios here on the slide. So each row that you're seeing when you look at schedules 1 and 2 or even 3, so each row is a unit operation, each column is a day. So if the day is filled with color, the unit runs that day. So let's first focus on scheduling examples 1 and 2. So having filling capacity on site can reduce risks and improve overall timeline, logistics, efficiencies. Logistics and scheduling can be challenging when you outsource or you're shipping product in between sites. There's cold chain maintenance and traceability that becomes quite critical. And then QC testing and batch release can be challenging if samples need to be shipped from site to site. Filling can become a bottleneck if you don't match filling capabilities with the batch output in the manufacturing schedule. So if you have a 5-day manufacturing process and a filling line that can handle a batch output of 5 days, so you would be able to produce about 55 batches per year with 1 day in between batches. So if you were able to decrease the filling time by 1 day to, let's say, 4 days, the number of batches would increase to 66, which is about a 20% increase. So now let's look at the third schedule example. So this one shows a multi-product scenario where you're manufacturing products A, B, C, and D. And the gray cells highlight the days when you have completed the manufacture of one product and you can clean, you can change over, you can prepare for the next product. So appropriate suite design and facility design are critical and will have an impact on how many batches and different products you can manufacture within the same manufacturing line. So room segregation will support efficient changeover and will also allow you to initiate cleaning and changeover of suites prior to the batch being completed. And single-use equipment obviously will increase flexibility, reduce cleaning needed in between batches, and will minimize that risk of carryover contamination. So the manufacturing platform, the suite layout, and the facility design all have an impact on manufacturing flexibility as well as multi-product facility capabilities. So now we're going to take a look at some of Cytiva's available solutions that support the mRNA vaccine manufacturing portfolio. So if you're not familiar with Enterprise Solutions, it's an end-to-end biomanufacturing platform and modular facility provider to the biologics industry. So from process equipment to platforms to automation to consumables to modular facilities and supporting services. So we provide end-to-end managed capacity from a single point of contact. So we have our two flagship products. That's the Flex Factory and the QBO. The Flex Factory is the flexible, configurable biomanufacturing platform. It's primarily single use, but it can be a hybrid. It can be a combination of single use and stainless steel, or it can be a combination of Danaher products as well as third-party products if there are gaps that still remain in the portfolio. And the modular facility solution always houses the Flex Factory. And on the QBO side, we have two options. We have the QBO facility and the QBO box. So the QBO facility is more of an external standalone manufacturing facility, and the QBO box is more of an internal biomanufacturing environment. So it's intended to be erected inside of a new or repurposed shell building or facility. And the foundation of all of our offerings consists of three elements. That's the supporting services, the automation and digital, as well as the fast track biomanufacturing services. So we know that there are three elements to the mRNA manufacturing platform, and Cytiva has solutions that support all of the segments in mRNA manufacturing. So let's first look at the standardized and modular solutions for plasmid manufacturing. So here we have a plasmid Flex Factory as well as a QBO box modular facility solution. So the plasmid solutions, they're cGMP compliant. They accommodate manufacturing scales from 50 to 200 liters, and they cover all the unit operations needed for plasmid manufacturing. And the plasmid Flex Factory always includes the equipment, primarily single use, the consumables, automation needed for plasmid manufacturing. And the QBO box always houses the plasmid Flex Factory, and it's designed to be flexible. It's designed to be multi-product, and it's easily expanded, and it's easily replicated. Now let's look at the standardized and modular solutions for mRNA manufacturing. Here again, we have an mRNA Flex Factory as well as a QBO box modular facility solution. As we look at the Flex Factory for mRNA, it accommodates manufacturing scales from 10 liters to 50 liters, and again, covers all the unit operations needed for mRNA manufacturing. We talked about what the platform includes, the equipment, the consumables, the automation. And again, the mRNA Flex Factory can be housed by the QBO box if customers want to go down the QBO path. So what do we think is the future of mRNA manufacturing? And do we think that it's here to stay? I would say most agree that we'll continue to have a demand for the pandemic vaccines. There are going to be uncertainties around, the future demand and what that will look like. We know that the virus has mutated. So will the current vaccines offer sufficient protection from the emerging variants? Will booster shots be required? And if so, what will the dose be? That will also have an impact on your manufacturing scales. Do we think that mRNA will be our new vaccine platform? And we know that the technology is fast, it's effective, it's been approved by the regulators. The race for new mRNA vaccines, targeting a variety of other diseases, it's already started, and it's booming. Then we see the growth of the oncology vaccine space. So mRNA has the potential to be a vaccine platform that spans billions of doses to, for the population, down to personalized therapies for a single patient. And we also need to remember that we're in the early stages of an evolving technology. We will see new mRNA types, new therapies. We'll also see improvements in manufacturing as new technologies and products designed specifically now for mRNA come to market. And even the already streamlined manufacturing processes for the currently available vaccines, they're being optimized, allowing the vaccines to be developed and manufactured faster and cheaper. So I would say mRNA manufacturing is probably a good investment for the future, and this technology is here to stay. We do know that we're likely to see significant evolution in the mRNA manufacturing space. So make sure that for your new capacity, it's flexible as well as adaptable. So I've shown that mRNA manufacturing, it doesn't have to be large scale to produce millions of doses. You should always stay flexible and fast with standardized and holistic platforms. You should always leverage that modularity and standardization for rapid deployment, maximize efficiency, and reduce risk with localized manufacturing. And do remember that there are flexible, there are modular, and end-to-end solutions available for mRNA manufacturing. So thanks, everyone. Thanks for your time today.